Welcome in this webinar about organoid technology and the ethical challenges. The webinar is co-organized by the EU-funded project Changer. It's the first webinar that we are organizing as part of Changer, and also from the non-profit organization BioMedLex. This is like the fifth webinar that we are organizing for BioMedLex. But today, it's a pleasure to combine both initiatives for this webinar. I have the real pleasure to have some very esteemed colleagues today, and I will present them to you a little bit later, but also have new people working in the field. So I'm really glad about having both junior and senior colleagues. If you allow me, I will make a very, very brief introduction, something like 5 minutes about the Changer project and how it is connected with organoids. And then we can start with our speakers. So I will share my screen. So let's have a look basically at what the Changer project is about and how does it connect to organoids. The Changer name comes from the challenges and innovative changes in research ethics reviews. The whole background of this call and the project, this is an EU-funded project coming from a Widera call, is the fact that we do have structures and frameworks in place that make sure that ethical standards and principles are being followed during research, and mainly this is through at the institutional or local level through the research ethics committees or RECs. And usually these are committees which are independent bodies, multidisciplinary, and they have the duty but also the responsibility to review research projects to ensure that they adhere to ethical standards and principles. However, on the other hand, although RECs have been proved very, very useful and as a guardian of research for more traditional biomedical research, however, nowadays, due to the emergence of new technologies, also new players in research, but new forms of research as well, we might have more complex ethical challenges related to research. So the question is whether the existing models of ethics reviews are fit for purpose in order to ethically assess research within the research ethics committees. So the overall objective of Changer is to explore and promote innovative changes in research ethics reviews. And these are mainly targeted to make sure that we can assess novel and disruptive technologies. But we would like to also bring on board researchers into this procedure. So we aim at what we call ethics by design in research. And we also aim to bring into a dialogue, if you want, both researchers and members of the research ethics committees through these innovative methodologies in order to be able to design more ethical, if I may say so, research. So I will not go through the description of the different work packages. However, it's important that we not only identify challenges and develop innovative approaches to address these novel and disruptive technologies, but at the same time, we provide training and develop training material for ethics experts, but also for researchers through this Changer project. So as you perhaps have also already guessed, organoid technology is one of these technologies, along with other technologies such as neurotechnologies or AI, that are kind of changing the research environment. and they raise more complex issues compared to more traditional biomedical research. We also have particularly sensitive issues regarding to brain organoids and embryo models. We will hear a lot about brain organoids in the coming talks. And it's also the fact that we create biobanks from these organoids that have some impact on how we should govern these biobanks. And as science progresses, we see that the complexity of organoids increases. For example, we might have assembloids, so these are more than one tissues which are used in a single organoid, or more than one organoid which are complex in a single system. And therefore, within Changer, we are interested in seeing how can we actually ethically assess research involving organoids. Because, for example, we might not be able to identify all the ethical challenges at the start of the project. And we need to go along and see whether, for example, organoids can become more mature and therefore raise different kind of ethical dilemmas during the research. And therefore, as part of Changer, we will have some training material developed for organoid technology specifically. So that's it from me as a brief introduction to organoids. And of course, we are very excited. We are at the 9th month of the project, so more is still to come. And we are a 15-partner consortium who are eager actually to work on these new methodologies and all the activities of the Changer project. So this is from me regarding Changer, and I hope that it was evident how this project connects to the rest of the talks about organoids that we will hear today. So before I introduce our first speaker, just allow me to let you know that there will be a Q&A session at the end of this webinar. So please post your questions along the talks, and I will try to either combine them or somehow address them all at the end of the webinar. Without any further delay, I would like to welcome Andreas Kurz, the first speaker for today. Thank you very much for accepting this invitation, Professor Kurz. It's really an honor for us because we will have the chance to hear about the science of organoids from real experts in the field. So Dr. Kurz is a geneticist with a PhD in cell biology from the Academy of Sciences in Germany. He has worked as an assistant professor at Georgetown and Harvard universities before becoming the director of the German Stem Cell Authority. He's a co-founder of the Berlin Center for Regenerative Therapies, heading the Stem Cell Research Lab with a focus on pluripotent stem cells and organoid research. In 2022, Professor Kurz joined Fraunhofer Institute for Biomedical Engineering at Berlin Institute of Health Charité to further data-related research and application in the stem cell field. His research concerns stem cell therapy and stem cell data management and ethics. He also coordinates the European Stem Cell Registry, which is an important registry for all those scientists using stem cells. And from 2017 until 2021, he was also a member of the European Group of Ethics in Science and Technologies. His main research concerns stem cell therapy and organoid research, as well as ethics. and therefore his talk today will be about the challenges of organoid applications. So Professor Kertz, thank you very much for being here. The floor is yours.
Thank you very much, Vasiliki, for the invitation and the kind introduction. So I will talk about organoids and how they are being made with a main focus on the brain, because that's today's topic as far as I understood. Also, my field of research in the organoid area is focused on the kidney and on the liver, not on the brain. But by preparing the presentation today, I learned a lot about this fascinating organ we all have, the brain, and how it can be used. So we all know that there are now many different types of organs which can be modeled in the shape of an organoid. But as I said, I will focus today on the brain, and we'll start with a few words about stem cells as individual avatars, biological and data avatars. Then move over to brain organoids, the status, how they're made, and which models they're being used for. Then how perhaps these organoids can be embodied. So basically taken out of the dish and exposed to the environment and react to environment and also signal to the environment. And about chimeras in this respect. So then we'll only talk about organoids derived from induced pluripotent stem cells or embryonic stem cells. Also, we know that organoids can also be made and are being made from biopsies, for example, and other cell sources. But for iPS cells, essentially, we have a patient or a donor taking any cell type from that donor, reprogram it, and make pluripotent stem cells. And these cells can then be used to develop organoids. And these organoids eventually can be, for example, put on an electrode array and signals can be measured, or they can put in a microphysiological system to expose them to drugs and measure the result of these drug exposures. And there are many other applications. We can also make from these pluripotent stem cells embryos or entities which look exactly like a human embryo or germ cells. And one of the main hopes for application of these cells and derived cell types and also organoids is, of course, cell replacement therapy. The other two also very interesting and hopeful applications are disease modeling and drug discovery. And of course, understanding or development of developmental pathways. So these iPS cells, these pluripotent stem cells, allow us to modify the cells, so essentially the donor genetic composition, genotype, and enhance that genotype, disenhance it, add phenotypic features or delete phenotypic features. So we can play around with this donor entity and its characteristics in the dish. And having these data available, we can compare that to the patient's or donor's own data and develop a model, an avatar, so to say, which we can experiment on in the dish. And that can be done, of course, not only for one person, but for anybody who is donating material. In this slide, I just described briefly how to make brain organoids, which is essentially very, very simple. We have a pluripotent stem cell colony. We put this colony into a format which allows these cells to aggregate and form clumps. They're called embryoid bodies, and these embryoid bodies then induce to form neuroectoderm. This neuroectoderm is placed into a hydrogel, and over time, over weeks, this hydrogel embedded neuroectoderm forms an organoid, which looks very much like for example, the cortex of a human brain or any brain. So these brain organoids on the lower right, you see how they look like in the dish. There is a clumps of cells. And when you cut them into pieces, then you will see that they are somewhat organized, self-organized into structure, which I will show on the next slide. And you can see who developed that technology first, Jürgen Knoblicht from the IBMA in Vienna. When you look into these organoids, you can see that they have the the cell types needed to form a brain. They have ventricular zone, they have a subventricular zone, they have intermediate zone, and they have a cortex layer. So the neural layer of the mature cells. And when you compare that organoid structure with the structure of a human brain or a mouse brain, you can see that it's relatively simple. It's not as complex yet. It is rudimentary in a way. And of course, the functions are limited. To make that more brain-like, adult brain-like, several things need to be done. The cell number and the cell diversity needs to increase. The synapses need to be pruned, and also synaptic connections need to be complex, more complex. The conductivity of the nerves must be increased, for example, by inducing myelination. And microphysiological systems, which I already mentioned, needs to be developed to modulate and measure the activity of these three-dimensional brain entities. So all the elements which we need to do that are already in this early stage brain organoid. The task is then to induce them in the right time, to shape them to the right place, and to make the brain organoid also more bigger in a way. For example, by assembling other different types of brain organoids from the frontal lobe, from the midbrain, from the brainstem, and so on. So when you compare the certain requirements or characteristics of the human brain of a computer or a laptop, you can see that in this table, which I took from a paper by Smirnova 23 from 2023, and you can see that there's a lot of speed in the human brain. It uses very low energy compared to a supercomputer and comparable to a laptop. The dimensions are, of course, much smaller than that of a supercomputer. The costs are, well, we don't know. There is no cost for a human brain in a human. There, of course, an enormous network of connections built in this brain. The memory is much higher than that of a supercomputer, and the storage capacity as well. So the organoid is somewhat not reaching that capacity of the human brain. We need to be clear, and that limits, of course, currently whatever we can or dream of doing with these organoids. So to improve the organoid, let's say, complexity, function, and capacity, researchers have developed a number of methods. Most importantly is turning from an unguided approach, the first, as I showed in the first kind of protocol, to a more guided approach, where we can form now forebrain, midbrain, hindbrain, spinal cord organoids, neuromuscular junctions, and other entities, and put them also together in multi-region assemblies to increase the function even more. In addition, we can use non-neuronal cell types or components, such as microglia derived from the immune system, blood vessels, to make this organoid even more natural and more, let's say, viable. These organoids can then also, of course, be transplanted. And when they are more complex, we can measure this complexity. I will go back to that part in the end of the presentation. So, and one of the key developments is now to use novel tools to measure exactly the size, the function, the morphology, the molecular composition, the metabolic capacity, and the functional capacity of these organoids in the different stages, different assembloids, and, and different sizes. And these methods become more and more, let's say, sophisticated and allow us now to standardize these organoids to make the findings more reproducible. And this is one of the key issues, not only for brain organoids, it's an issue for all types of organoids to make the work done with them reproducible. Standards are being developed, or the ethical standard to increase this reproducibility. The next part of the presentation will go into the application of these organoids. And one of the major applications is, of course, disease modeling. And this is possible with these organoids, as has been shown for a number of neurodevelopmental disorders like autism, microcephaly, Rett syndrome, of neurodegenerative disorders like Alzheimer's, Parkinson's, or Huntington's disease, psychiatric disorders, depression, schizophrenia, bipolar, and others, and for the modeling of brain tumors. And the data of these models show that the diseased and the standard phenotypes can be adequately mimicked in these organoids, even at this relatively simple premature stage on the cytological, molecular, and functional levels. The example here, also from the same paper from Smirnova, shows the use of the comparison between healthy and Alzheimer's organoids, where it's obvious how different the myelin sheet wrapping around an axon looks like in these two nerves in these organoids, and how disintegrating the microtubule with tau protein tangles appears compared to in the Alzheimer neuron compares with the non-Alzheimer neuron. So, and this can also be done with other diseases. And the number of diseases being modeled with brain organoids goes so far that now the first biobanks are being developed to make organoids from a number of especially psychiatric disorders, which are very difficult to tackle and to diagnose. So another application is computing. The idea is to use, instead of a silicon chip, a brain organoid to manage computing tasks. So what you essentially do, you generate an input, translate this input in an electrical stimulation on a spatial temporal level, and expose the organoid to this input. Then the organoid hopefully forms synapses, runs synapses, and establishes a network which can recognize this input. And then this activity of the brain organoid can be decoded again with a, for example, electrode array, and the output can be displayed. So this brain organoid-based computing is advancing quickly. What we can see here and this is a paper from Tsai et al., is how they do it. Essentially, they put a brain organoid on a multi-electron array chip, and this system is then stimulated via the chip. So you can see how the organoid on this chip looks like, and ideally, you get some kind of learning by the brain organoid with this signals. So if there is no organoid, you don't see anything. If you block the synaptic plasticity, you also do not see any changes in this case, synaptic reshaping. But if you train this organoid, you should get a reproducible synaptic modulation, modification, pruning, and connection network building in this organoid. It's called ONN, Organoid Neural Network, what's established here. And what then can be done, of course, with these neural network or organoids, you can measure organoid intelligence, it's called, by using these organoid for AI function. You have an output from these organoids, for example, in this kind of measurements, which I just showed. Use another AI, map this output, and then identify certain responses from that output. What can be measured from these organoids is, for example, neuroplasticity, for example, the connection strengthening or weakening. You can characterize the responses, for example, based on calcium signaling, and map these responses. And you can then, of course, use that for drug screening to see whether your model is changed in certain drugs in a way which, for example, reduces the Alzheimer phenotype and increases into a more, let's say, standard response, or delays the Alzheimer phenotype development in a brain organoid. One input could, for example, be speech. And then the paper of Tsai et al., which I already referred to, speech was indeed encoded, and this was, in this case, Japanese vowels, encoded in a spatiotemporal sequence in this array, in this microelectrode array. The brain organoid was trained on this array with the vowels, and then it was tested. This is a training schedule, so there are 2 days training, and after 2 days the goal was to distinguish the Japanese words from other speakers. And what happened is indeed that you could see this difference. So this is a chance output, and this is the output after training. The accuracy over training epochs or training time should increase. And that was indeed what happened. For example, this is strengthening of the synaptic connection and weakening of other synaptic connections. So pruning, essentially pruning, and both effects increased over time. And what you can then see after stimulation of this trained brain organoid is indeed a kind of recognition of the Japanese vowels compared to others. So this is essentially a simple translation of a auditory input into an electrode and train the organoid to recognize this input and measure it as an output. Similarly, one can imagine, of course, other inputs, and one very interesting input could be a light input, an optical input. A few years ago, brain organoids have been developed with optical cups are emerging from them. These are somewhat light sensitive and signal into the brain, into this brain organoid, a sensory signal which can be measured. So if we think further about this, it could be possible to use that also to train organoids on light signals or optical signals. So how else can that be used? And there are now a number of companies jumping into this prospect of using the brain organoid either for computing or to learn certain tasks. So one of the platforms is called, or one of the, it's called Final Spark, a neural platform where in this case, 4 brain organoids, each consisting of 10,000 cells, are wired to a biocomputing array. And this biocomputing array can then measure input on these brain organoids. This other one is not really an organoid, it's a more two-dimensional, 2.5-dimensional neuronal carpet on an electron array. But this carpet has an interesting capacity to learn a game called Pong that was published a few years ago. Whether it really learns that is still in debate, but it shows that you can train these neurons even on a two-dimensional platform to perform certain tasks. And before I come to the chimeras, I want to show this slide where the output, so we can modify the input, we get a certain output. The output can be utilized, in this case, to move a robot. And this robot essentially contains a brain organoid or a neural organoid, which is enclosed by a chamber which contains everything a human brain would also need: a fluid, nutrients, temperature control, and a protective casing. And this organoids then moves, so to say, the computer upon a certain learned stimulation. And this was done in China at Tianjin University. And one can imagine then if one combines that kind of system, which is looking autark, it's of course not autark, but looking autark, with a feedback mechanism which signals to the brain organoid, okay, now I need more nutrition and it can feed itself, then the embodiment which you see here in a way which looks very close to a human, it's not, which the embodiment could then lead to a self-awareness of this brain organoid. But there's not only the only, it's not the only way, it's not the only way to, let's say, functionalize this brain organoid and use this output to, for example, trigger a movement. You can also use a more natural host to do that. And this has been done, is increasingly being done using animals and transplanting human brain organoids into animals to test, of course, their functional connection into a damaged brain area. And in this particular case, the visual cortex has been damaged in this case in an adult rat and replaced by an organoid, a human organoid. And then it was measured whether this organoid can react to light stimuli to the rat, and it indeed can. So it's integrated. It was integrated into the rat brain and the rat then reacted to that transplanted organoid by orientation selectivity. So this is my short introduction into the area of brain organoids. Thank you very much to giving me the opportunity to myself learn a lot about brain organoids and present it here.
Thank you very much, Professor Kurz. I think that we all learned, and for me at least, you explained it very well. You explained complex things in a more simple way, so I think that it is very useful to understand the different potential abilities that brain organoids might already have or might even have in the future. So thank you very much for this. We're now moving on to our next speaker, Professor Andrea Lavaggia. He is an associate professor of moral philosophy at Pigasso University in Italy, and he has recently been included in the Stanford Elsevier's top 2% scientist list for 2024. Andrea's research focuses on the ethics of new technologies and neuroethics specifically, and he has written a lot about the ethical challenges of brain organoids. So his talk today will be about new biological entities and their status. Professor Lavaggia, thank you very much for accepting our invitation. It's a pleasure and honor to have you here, and we are all ears and looking forward to your talk.
Thank you very much for the invitation and for the kind presentation. I hope you can see my slides.
Yes, but yeah, in full screen now. Thank you.
Okay, thank you. I assume I have 20 minutes.
Of course, yeah.
Thank you. Thank you once again for having me today. It's a very significant event, and I'm very happy to be the second speaker after the great presentation. Before me, it was very, very effective in showing what organoids are and what they can do now and probably will be able to do in the future. So my presentation will be focused on some ethical issues surrounding brain organoids in particular. So the general idea, considering an organoid as a solution, as probably we can agree on, the question is if all is going well or not. And then we can consider that human organoids in general can be considered a more acceptable form of experimentation that than that of human fetuses, animal, and obviously adults. And in this sense, no living being is destroyed, damaged, or put at risk. And probably we can also have a reliable level of outcomes from these new means of experimentation. And again, organoids can be cheaper, both in terms of material and also working hours. And so this is another good reason to consider organoids. But I think that we need to be careful in celebrating, so from an ethical standpoint as well. So we need to have some caution in this approach. And now we can obviously consider some issues which can be divided into main categories, extrinsic and intrinsic ethical issues. And so these are some of the classical, if we can use this adjective, issues in bioethics concerning the biomedical field and new strands of research related to human tissues and so on. And so we can consider all of these types of issues. Obviously, we do not have time to delve into them, just a list in order to have an overview of the potential issues that can arise using or resorting to organoids. So we have issues related to the donors, and we can have different potential solutions and other concerns that can be addressed yet. And it goes the same way for other issues, informed consent, privacy of genetic information, intellectual property, and also earning distribution. So maybe in the Q&A session, we can say something about specifically this aspect, but I'd like to focus my presentation on another topic related to brain organoid, maybe something which is not now so pressing, but that it can be the most relevant issue in the future. And so I use this quotation from Hank Greeley, and he wrote about a dilemma, and finally, the point is, if it looks like a human brain and acts like a human brain, at what point do we have to treat it like a human brain or a human being? So this is a important question. Obviously, these days we do not have brain organoids in the lab which are conscious or which should be treated like a human brain, but the point is how far are we from that point? And so this is the introduction to the topic. Very briefly, we can also consider a very controversial study, probably well known to all those who are involved in this field or are cognizant of the organoid research. In this study, scientists have shown that brain organoids generated by induced pluripotent stem cells can develop periodic and regular oscillatory electrical activity that resembles the EEG patterns of premature newborns. And this means, very briefly, that 10-month-old organoids, even in absence of external or subcortical inputs, can develop according to a specific genetic program typical of all humans. And the most surprising feature of the study is that an expert software trained with the EEG data of preterm infants was able to evaluate with a good approximation the age of cerebral organoids based on their electrical activity. It means that from the point of view of the software, organoids and preterm babies were indistinguishable. Obviously, it does not mean that organoids were conscious. We can rule out that, but it is very significant, in my view, that organoids and preterm babies had similar EEG data at that stage. So we now have also new methods of cultivation of cerebral organoids that have allowed to generate diverse nerve tracts with functional output. And in this sense, cerebral organoids have proved capable of inducing movement, although not yet a purpose-oriented kind, obviously. Again, there are other studies, but I do not delve into them, as before we have seen all the new advancement in this field. So we can have for granted that organoids are capable of displaying features typical of the human brain. So it's important, in my view, to consider also what scientists think of these ethical issues. And a colleague of mine and I conducted a survey recently published with a group of scientists involved in organoid field, and then we got these general responses, and as you can see, in general, scientists are skeptical about the idea of consciousness emerging in brain organoids today, and they are quite skeptical also in the possibility that organoids will be conscious in the future. And also considering moral status, they said that there is no ethical relevant difference between organoids and other tissue products. So in general, scientists working with organoids are not concerned with specific or particular ethical issues in their field. And I think this is something we should take into consideration. Considering instead the idea of the potential emergence of consciousness in organoids, I think it's important to propose some definition and some ideas, precise ideas on what we mean by sentience and consciousness. So my take on sentience is that it is the minimum degree at which a living entity can feel something, whereas consciousness is the phenomenal and so higher level at which an entity can be self-aware as well. So you can distinguish these two level of being conscious in the broader sense, but obviously some scholars, as you probably know, use this term in other ways. So it is a matter of consensus. I propose and submit these definitions. And it's important also to note that some scholars hypothesize that this emotion, the basic emotions we can think about in animals, for example, are experienced in a certain way only thanks to the sophisticated processing typical of the nervous system of mammals. So it means that organoids which are not perfectly identical to a human brain cannot feel and think as humans do. But it's not implausible in other scholars' view to suppose that a living being can feel pain and also experience lack of air, for example, even without having an inner model of the world of itself. And it means then specific entities, not organoids in this case, but in general, can feel something, can be sentient, even they are not similar to us concerning the brain and the nervous system. So this is our hypothesis, but it is important to consider that. And so moving to the potential presence of consciousness in human cerebral organoids, we need to ask two questions, essentially, in my perspective. The first one is an ontological, ethical one, in a way. It concerns how to identify what consciousness is and what characteristic attribute moral status to an entity. And the second one is a more epistemic, so to say, and it concerns how consciousness can be found and evaluated, and the characteristics that allow moral status to be given. So we need probably to work on these two strands of investigation in order to have a good ethical overview on the possibility of development of organoids in the future. So very briefly, possibility to evaluate the emergence of consciousness is due to some indirect ways of investigation, because obviously brain organoids do not display behavioral outputs so far. And so we can consider the neural correlates of consciousness as a marker of the presence of consciousness in a way. But again, we have a big problem because we have different theories which can account for different neural correlates. So we can only speculate, and very briefly I show you this table in which we can see different theories and different tenets of these theories, and how these tenets can fit with the idea of organoid consciousness. And if we can try to make an evaluation, we can exclude two of them. It's impossible for organoids to be conscious if temporal spatial theory and body theory are the true theories of consciousness. It is possible that some organoids is conscious if we think that the integrated information theory and the neural workspace theories are the true theories. Obviously, they cannot be together the true theories, because we know they have very different principle and tenets, but these theories are compatible in principle with organoid consciousness. So this is the theoretical framework we need to consider in this sense. Obviously, there is a very, very hot debate on this topic. Just to let you know, some of the most recent paper and claims on that. So if someone is interested in this debate, which is both philosophical and ethical, can look at this paper concerning this very important debate. So, going to the conclusion, obviously we are interested in the possibility of emergence of consciousness because sensing consciousness are deemed relevant for the attribution of moral status to an entity, and in particular to a biological entity. And so some scholars proposed a precautionary principle, something which is very relevant in that it requests very tight restriction in research if we adopt this principle. And obviously, in general, if we want to introduce new rules, new guidelines, we have repercussions in research. So I need to skip some part due to time reason, but if someone is interested, I think the organizer can have a way to send or to make my presentation available for the audience. And so I'd like to conclude with some words on moral status as such and moral value. We know that moral status in general is a condition for which a certain entity receive consideration in the ethical sphere for being that entity and not only for its relation with other entities. So intrinsic value in a way. So we can also have this pyramid of moral status with different levels. And in general, scholars and philosophers do not think that moral status is equivalent to moral value, because the former is a basic condition that does not determine the degree of moral value. But we obviously need to start with moral status, and granting moral status is something not always easy to do, because obviously we needed to detect subjective interest which are linked to certain subjective experiences. So to obtain moral status, it is in general necessary to have some form of sentient consciousness, as said before. And so moral status can be attributed based on various justifications, but the most relevant, the most important is the presence of sentient of consciousness. So concerning human brain organoids, the point is if they can be sentient or even conscious, obviously they should be granted moral status. And this is the main point. And just to conclude, a quite interesting proposal from Christoph Koch, who is a very relevant figure in the field of consciousness, and brain research, and he proposed to consider brain organoids very high in consciousness dimension and very low in the intelligence dimension. So distinguishes these two dimensions. We can have obviously human beings as they are the highest entities in both dimensions, but probably we can also consider the position of organoids in this framework, and probably Koch, in my opinion, is exaggerating in putting organoids so high in consciousness so far, but probably in the future it's possible that some organoids grown bigger with specific features can be sentient, and at that point we obviously would need to decide what to do. So my final point is just some question. It's curious that I too have the same robot guided by an organoid. And so finally, I said some question in order to think about these ethical issues. What should we do if cerebral organoids presented a glimmer of consciousness in the form of integrated information? I say that because probably integrated information theory is the theory which allows organoids to be conscious more easily. And then we will be able to use brain organoids to try to unravel the mystery of consciousness. This is another very pressing question in research, because in the future this is a possibility that some scientists can take into consideration, and then do we need new and special rules for the creation and use of human cerebral organoids, which is obviously the main question at the center of this event. And thank you very much for your attention.
Thank you very much, Professor Lavazza. We do have some questions that we will address at the end of the talks. So thank you for the ones that have already posed some questions, and we will get back to them. So I will now move on to our third speaker. We will move on to Elasmar Hossam Hessam Ali. He is a legal professional and a master's student in bioeconomy, biotechnology and law at the International Hellenic University in Thessaloniki. He is currently working on organoid intelligence as part of his master's thesis, and his talk will be about the notion and evolution of organoid intelligence. So thank you very much, Hossam. The floor is yours.
Thank you very much, Professor Malaki and BioMedLex and Changer Project for the invitation. Thanks for having me. I would like also to thank Professor Lavazza and Professor Kurtz for their insights during their presentations. Very integrated. So let me share my screen, and then I'll kick off my presentation. Can you see it in full screen? If you
yes, everything is fine.
Right, perfect. So my topic here is about the organoid intelligence, the notion, and the evolution. So basically, I'll go through the agenda very quick. We'll go through the introduction, and then I'll touch on the organoid intelligence versus neuromorphic computation, where they correlate. Also, next we'll move on to the progress and advances in organoid intelligence. Obviously, some parts in this respective point have already been covered in the previous presentations, so we'll not take so long on these. And then I'll move on to the potential ethical concerns related to OI, and then we will conclude. So to start, just to note that the use of brain cell cultures for robotics and learning tasks have already been around for nearly 2 decades now, a little over 2 decades. In 2001, study by Schaaf and Merrim showed that the rodent primary cortical neurons can learn by responding to low-frequency stimuli, resulting in distinct post-learning electrophysiological patterns. And the key difference is that the human brain obviously differs significantly from rodents like rats and mice in terms of the size, complexity, and pseudo-architecture as well. Then another major turning point was back in 2006, when Yamanaka discovered or developed the induced pluripotent stem cells, marking a pivotal moment in the stem cell research. And that point helped revitalizing the field of stem cell research. And next, we underline another milestone back in 2013, where Lancaster developed a method for creating cerebral organoids from induced pluripotent stem cells, and that has enabled the study of human neurodevelopmental process in vitro. Now such progress in the stem cell culture and bioengineering studies has elevated the sophistication of cell cultures to mimic the in vivo physiology and structure, forming what is referred to as the MPS or microphysiological systems, and that comprises in vitro organs such as organs on chips and tumoroids and organoids. One key thing that I would like to point out here, obviously we've discussed on the organoids being a miniaturized self-organizing versions of organs or tissues that replicate their in vivo counterparts, whether be it kidney or be it a human brain. But there is like a clear distinction that that needs to be defined here because of the interchangeable use of terms. Terminology here matters because we've seen terms such as cortical organoids, cerebral organoids, brain organoids. What's the difference? For example, cortical organoids are a type of neural organoid characterized by a deficiency in oligodendrocyte progenitor cells, while the brain organoids is a more general and encompasses both cortical organoids and cerebral organoids, which are region-specific brain organoid models. I bet we've seen this slide before. Obviously, the use cases of brain organoids, so I wouldn't spend much time on this. We can use brain organoids, obviously, for disease modeling and understanding human brain development, drug testing, and neurodegeneration to research neurodegenerative diseases such as Alzheimer, Parkinson disease, and ALS. And yeah, obviously this is not new anymore. We've seen that before, and it's not science fiction, but it's basically, if you're familiar with brain organoids, that might be the first thing that you will imagine, or it will come to your mind when we say organoid intelligence. You will just imagine a humanoid or a robot like this one with an organoid inside that's running the whole thing, powering this system. And here is where things intersect. We have a brain organoid, and we're trying to integrate it with AI, and this is where OI comes into play. So the overarching aim of OI, as defined by Harting et al. In 2023, aims to harness the computational power of biological neural networks to create novel forms of intelligence and biocomputing. Now, looking at the term organoid intelligence, we've just defined it being with the aim to integrate brain organoids with computer technology for biocomputing and synthetic intelligence. The term OI, organoid intelligence, abbreviated as OI, is used for this approach to stress its complementarity to AI. With AI, we know that the aim is for the computers to perform tasks usually done by the human brains. So similarly, the term OI was coined to mimic the same approach, but on a different scale. The term organoid intelligence was first coined in 2022 as part of the Organoid Intelligence Workshop held at the Johns Hopkins University. And it was followed by the Baltimore Declaration, which basically outlined the potential legal, ethical, and societal challenges that might come to the way of this new emerging field. And obviously, that was the first point to lay out or to pave the way for the consecutive paperwork that started using the same term, organoid intelligence. Now, another key question, why is organoid intelligence? Why do we need organoid intelligence? What is it trying to address? And now, to get to the answer for this question, we need to understand the human brain capabilities and compare it to the supercomputers. Now, supercomputers, they require significant space and power. And we come back to that point where the frontier supercomputer, this is the most potent supercomputer at this point, where it processes over 1 quintillion operations per second, and it occupies 680 square meters and needs 22.7 megawatts of power. In contrast, if we look at the human brain, it operates with only 20 watts of power and weighs circa 1.3, 1.4 kilograms, achieving the same rate of operations. One thing to point out as well is that organoid intelligence is becoming more of a key investment area, and we see the National Science Foundation in the US investing almost $30 million for the fiscal year 2024 and 2025 next year Just for this project that supports foundational and transformative research aimed at enhancing the design, engineering, and fabrication of organoid systems that can dynamically process information and interact with non-living systems. Okay, so we just mentioned the neuromorphic computing or supercomputers. What are they? Basically, the neuromorphic computers are supercomputers built on two types of neural networks. First is the artificial neural networks, or ANNs, and these are a series of algorithms that run on conventional computers to simulate the logic of the human brain functions. And there's another type of neural networks, and these are called the spiking neural networks. And these spiking neural networks try to attempt to replicate the functional principles of the human brain and communicate through spikes. Obviously, the artificial neural networks have been around for quite some time. The spiking neural networks are kind of more recent. In terms of trying to mimic the human brain structure or architecture to create computers, this basically relies on two fronts. The first is using or relying on quantum computing, and the second is the neuromorphic computing. The quantum computing, we're not going to delve into this. It relies on quantum bits or qubits, and these are basically like the conventional logic, the zeros and ones in the standard neural networks, which are being used in the computers. The only difference is that they have the potential to lose data if these qubits change their status. It's quite complex, but we'll focus here on the neuromorphic computing. And the neuromorphic computing, it refers to the non-von Neumann computers, just unlike the conventional computers that we're using now. The von Neumann computers, they have separate processing units and memory units for storing data and instructions, while neuromorphic computers integrate processing and memory within neurons and synapses. And this is a reductive illustration of the difference between the structure on both the von Neumann architecture and in neuromorphic computation architecture. A very prominent example of the neuromorphic computing, it was brain scales. This has been part of the Human Brain Project funded by the EU between 2013 and 2023, and resulted this supercomputer, which is based in Heidelberg in Germany. A very recent advance in this sphere is Deep South, and this neuromorphic computer has been developed by researchers at the International Center for Neuromorphic Systems at Western Sydney University, and it's proposed as the world's first supercomputer capable of simulating networks at the scale of the human brain. It uses neuromorphic system that mimics biological processes, achieving around 228 trillion synaptic operations per second. So far, there aren't many information around the efficiency of Deep South in many other aspects like power and so on. It's very recent. It's just launched in Q1 2024. Maybe it's too early to decide, or to at least to go through the updates of the system. So that brings us to, where do they fall short? Where do neuromorphic computers fall short? And this is where OI comes in. Although the neuromorphic computers have made significant strides, but they still fall short in certain aspects, like in techniques like weight quantization, which can reduce memory and computation needs for embedded systems. However, if these are not applied carefully, they may compromise the network accuracy. One other thing is that the neuromorphic systems, they often depend on conventional machines for their software architecture, which can reduce their advantages. Also, the development of neuromorphic algorithms face challenges due to lack of clear benchmarks and metrics, as well as insufficient programming abstractions for non-machine learning algorithms suited for neuromorphic applications. In terms of the advances in organoid intelligence, just to stress on these advances, we will go through some of them, not all of them, because the majority have already been covered in the previous presentations. Dish brain, we've seen that before. It's basically this system that was developed by Monash University in Australia and spun off to Cortical Labs. And it was evident that it showed apparent learning within 5 minutes of gameplay for Pong. And that was not seen in control conditions. The system architecture is still the same, just an organoid and electrophysiological setup, MEA, multi-electrode array, and the organoid. And this is the same as the next model, which we've already seen as well in the previous presentation. So I'm not going to spend much time on this brain aware. It definitely had advantages, and it had very good at speech recognition and nonlinear equation prediction, but it had some issues in terms of the proper maintenance and support to optimize computational power. And even though it had lower power consumption, there are still peripheral equipment like the CO2 incubators and computers that consume significant power. Final Spark, the model of Swiss startup Final Spark, we've seen that before. It's available for everybody to try. I think it's already there at a cost. So yeah, and this champ, the Meta BOC. So this is the third time we've seen this. It's been already developed by Tianjin University with the support of the Southern University of Science and Technology. The organoids in use in this model, they were cultivated using low-intensity focused ultrasound simulation, which is believed to enhance the foundation for intelligence development. And there are even some videos around for the system, the Meta BOC, where the brain organoid runs the robot, and it autonomously controls the robot for tasks such as the obstacle avoidance, tracking and grasping, and completing inspired work for various brain-like computing. It's very interesting. And there's this example of this P organoid, and it's quite nice. It's trying to resolve the mechanism behind music-induced cognitive changes, which have been largely unknown. Basically, it introduced this Pi organoid library for simulating organoid learning models with machine learning techniques. It relied on historical data from actual individuals, like EEG data and functional MRI data, and they compared it with the outcome of the organoids. And it seems that it will this model provides insight into music perception and cognition, and highlights the value of synthetic models in neuroscience research. It's a very recent study. It was published just only in July. So moving on to the ethical concerns. So here at this point with OI, there are two aspects to consider. The ethical aspect, obviously the ethical and legal aspect of using the brain organoids employed in the systems. And there is another aspect of the ethical issues of using AI. We've seen in the ethical issues of using brain organoid, we discussed a bit about the consciousness and moral status. There are other issues relating to informed consent and human-animal chimeras and commercialization. On the other hand, there are still issues to be resolved in terms of privacy concerns, environmental impact, and autonomy and control. Now, organoids, when they are being integrated into systems like those we've seen before in the previous slides, they become AI-enabled organoids. And on this front, they can, these capabilities that we have here listed through machine learning, we would need AI for, this meta BOC model enables brain organoid to autonomously control robots for tasks such as obstacle avoidance and tracking and grasping, completing inspired work for various brain-like computing. This is a very interesting video for this for how this model is being used. And this is the last example. It's a very interesting one. A project called Organoid. And basically what it does is that it aims to resolve or understand the mechanism behind music-induced cognitive changes, which has remained largely unknown. So this model introduced the Organoid library for simulating organoid learning models with machine learning techniques. It's a deep organoid learning model using something called bidirectional long short-term memory networks, and relied on historical data from EEG responses and functional MRI from actual persons, and tried to connect these with the results from the brain organoids. And it provides insight into music perception and cognition, and highlights the value of synthetic models in neuroscience research. So the last point that I wanted to underline here is basically the ethical concerns behind organoid intelligence, and this is split in two parts. The first is the ethical and legal issues of using brain organoids. We've seen the moral status and consciousness in the previous presentation, also the informed consent and the human and animal chimeras and commercialization. On the other side, there are certain ethical or potential ethical issues of using AI, such as the privacy concerns, environmental impact, and autonomy and control, and the accountability for AI errors. So once we have an organoid and it's connected or interacting with AI, then it becomes an AI-enabled organoid, and we see certain uses for AI in this context, like machine learning can guide the structure of discerning of the matrix gel and the fine-tuning of cell culture conditions, and it also helps with the identification of active inducing factors, also the assessment of external stimuli, and the analysis, the image analysis of morphology scale and organoids scale, and the image analysis in tissue scale. So yeah, basically for the accountability and liability, identifying the responsibility when an AI system makes errors or causes harm still challenging. It's crucial to establish clear lines of accountability. On the environmental front, the computational resources needed to train and operate AI models can greatly affect the environment. As we can see, there's this MIT technology review reporting that one AI model can emit more than 626 pounds of carbon dioxide equivalent, which is nearly 5 times the lifetime emissions of an average car. So to wrap up, engineered brain organoids are advancing quickly, and they're enhancing their functions with significant potential across neuroscience, medicine, computing, and robotics, and brain-machine interface. And the ethical and regulatory challenges, such as the accountability and AI errors and the data privacy, are of paramount importance, and these have to be subject to societal discussions on different fronts, bioethics and legal as well being involved. So yeah, that brings me to the end of the presentation. I give the floor back to Dr. Malaki to proceed.
Thank you very much, Hossam. I think that we can see how complexity progresses, as science progresses, also the ethical challenges also progress. So thank you very much for this presentation. So we have the pleasure to have with us Panagiotis Kavouras. He has been working on a project called the Hybrida project, specifically on organoid research and the ethical aspects of organoid research as well. So Panagiotis Kavouras is a senior researcher at the School of Chemical Engineering at the National Technical University of Athens, and also a researcher at the Institute of Health and Society, Center for Medical Ethics at the University of Oslo. He is a physicist, so you can see that we have an interdisciplinary panel today, with a master's and a PhD in physics. For several years, he had been involved in research on the synthesis of glass ceramic materials with tailored properties and on the characterization of physical and engineering properties of various types of materials. During the last 7 years, he has been focused on research integrity through his participation in several EU-funded projects about research integrity. Panagiotis today will talk to us about this project, which was specifically aimed to, among others, I would say, to try to explore the ethical challenges of organoid research. And today he will present to us some concrete guidelines that came out of this project that has finished relatively recently. The title of his talk is the Hybrida Operational Guidelines and Code of Conduct for Organoid Research. So Panagiotis, thank you very much for being with us today. I think that it's very interesting to see people that have been working on actual, or not on actual organoids, on the ethics of organoids for the past years, and the guidelines that came out of this project are very useful to everybody working on such entities. So the floor is yours.
Thank you very much for the introduction, Vasiliki. As you realize from my expertise, being a physicist, I'm the lucky one who will present some parts of the work that has been done by many other scientists, members of the Hybrida Consortium. And before I start, one disclaimer from my side, I do not have the photograph of the organoid and the robot. So in this respect, it will be a bit different from the previous presentations. It was, of course, just a joke. It is a real honor to follow with this presentation the very esteemed presenters before that were present in this nice webinar. So what is in a word? Hybrida project comes from the word hybrida, that is a person born of a Roman father and a foreign mother, or of a free man and a slave. So this kind of hybridity in this kind of historical context could be also said that is reflected also to the consortium that is composed of bioethicists, people, experts in research integrity, philosophers of science, historians of science, and of course, experts in organoids and organoid technologies. So the aim of that project, it is of course easier to say than to do, is to develop a comprehensive regulatory framework for organoid research and organoid-related technologies. And we started at the beginning of 2021. The project has ended a few months ago by studying with 3 mapping work packages 3 types of uncertainties. Firstly, the conceptual uncertainty, whether organoids could be considered to be persons or things, on epistemological uncertainty, and more specifically, what type of uncertainty do we have for organoids? Is it quantitative or qualitative uncertainty? Or we are at a place that we are completely ignorant or quite ignorant of what an organoid is. And third, regulatory uncertainty on how to merge regulations dealing with persons and things. So In a sense, we were trapped in the perfect storm. We had to study a very novel technology that raised research ethics-related issues. We yet were not certain if they were explicit or novel for organoids, or there were ethics-related issues that were also recognized in other technologies. That was the case, at least when we had finished this mapping. There were also issues of terminology, pinpoint which were the relevant fields of research, and of course, issues that had to do with communicating to the general public of that type of technology. So we used for the outputs of Hybrida, one of these is the code of conduct and the operational guidelines, is that we followed a three-tiered co-creative process, beginning with public workshops, 3 deliberative workshops that took place in Denmark, Greece, and Italy, where 51 participants in total shared their views, their worries, how they see trust in science, more specifically for the biomedical field in general. And then at the second stage, with 2 expert co-creation and consultation workshops and 15 expert interviews that provided input to the first version of the Hybrida outputs. And then at the third stage, with 6 focus group interviews and 35 in total leading experts to have the assessment and validation, and at the end, create the final version of Hybrida's outputs. So we could say that the 3 different types of outputs of Hybrida respond to 3 questions. Why there is a need to enhance existing ethics and regulatory frameworks, what standards of conduct and good practices we should follow to be in line with enhanced ethics and regulatory frameworks, and how to actually implement these new standards and good practices, not only from researchers, but also from members of research ethics committees, of research integrity bodies, people that work in funding agencies, so not only targeting researchers. So these types of questions, starting from the base of that pyramid, we are yet quite distant from the laboratory, and we go to actual hand-on guidelines for researchers and these 2 other types of stakeholders. So going to the operational guidelines, a very brief overview, of course, because we have a very strict time limit right now. So they are recommendations to organoid researchers that are designed to streamline certain working procedures according to best practices. And they aim to help researchers ensure reliable research, development, and production of organoids. They are structured in 4, let's say, sections with quite interesting acronyms. The first is the MEOW, then the ECHOS, the RICOCHEC, and the TRUSTED. So I will very briefly present them. So the MEOW part of the operational guidelines target researchers, and that section provides them a way, a decision tree, if I may say, for them to be able to present the necessary information when they, for example, publish a paper or when they communicate with their peers, and also metadata that are related to regulatory aspects. It is not just a list, but we have created an online questionnaire. You see some screen captures that is very helpful for researchers as a memory guideline for people, for things that have to be reported in the context of their work. The MEOW is echoed for in another section of the operational guidelines that targets experts and scientific committees that must be well equipped to evaluate the quality of organoid description when someone asks funding for such a kind of technology. And then these echoes find a place at RICOCHEC, that they are operational guidelines for research, for members of research ethics committees and research integrity offices. And we know that there are cases that the research ethics committee or a research integrity body might not have experts in organoids, or they cannot find advice from someone expert. So with this kind of help, experts are not necessarily needed, but at least there is some kind of guidance for these bodies to be able to assess this kind of grant applications or running projects. And of course, the trusted part that has to do with the type of informed consent that Hybrida suggests. Professor Lavazza mentioned about the issues that have to do with informed consent in the organoid field. What we propose is the dynamic consent and the consent for governance model. In this case, actually, the researcher goes back to the donor and asks her or him if he accepts the tissue that has been donated many years ago, perhaps, it can be used for a type of research that was not anticipated when he gave the consent. And the consent for governance model is the same. It is just the researcher and the donor. You have an intermediary, an intermediate entity that does that work, going back to the donor, if possible, of course, for a renewal of the consent. I will just present a list of the things that are present at the Code of Conduct that aims to develop and support research integrity within an ethical research ecosystem. We see we have sensitive domains of research integrity that are more specific to organoid research, good research practices, again, specific for organoid research, guidelines that have to do with violations of research integrity, and with the engagement with stakeholders that we believe, and we saw that at the public deliberative workshops, that it is very important and a very challenging thing to do for a researcher. And this is my last slide. I'm at 10 minutes right now. We are, in addition, suggest a supplement to the European Code of Conduct for Research Integrity. That was one of the contractual obligations of Hybrida, to see whether there is a need to suggest something additional to the European Code of Conduct. And the one additional thing that Hybrida suggests to be included has to do about the responsibilities of researchers that are relevant to human biological samples and associated data. And one of these fundamental principles of which is respect for the donor and for their consent. So this is our suggestion to be added to the European Code of Conduct for Research Integrity, something, a high-level document that I think most of you are very well aware. Thank you very much for your attention. These outputs, these reports are freely available on our website, with another small disclaimer that we are waiting for the review of the project to be done in less than a month. And as soon as we have finished, and I believe until the end of that year, or the latest, the beginning of next year, these reports will have been finalized, and everyone will have access to these finalized versions of the deliverables. Thank you very much for your attention. I would be very willing to respond to any kind of questions in written form or orally. Thank you again.
Thank you very much, Panagiotis, for being short. I realize that this is a constraint, and we're looking forward to all the outcomes of the Hybrida project. So let me now quickly move on to our next speaker, who is Natalie Evans. And I know that she hasn't got too much time available, so I will briefly introduce her. Natalie Evans is a senior social scientist with a background in public health and anthropology. She has been involved in many EU-funded projects that have to do with ethics and research integrity. And she is a co-founder of the Embassy of Good Science. And this is what her talk will be about today. She will present us the Embassy of Good Science and within the Changer project, we have the pleasure to collaborate with the embassy, which is an online open go-to platform. So thank you very much, Natalie, for being with us here. The floor is yours.
Thank you so much, Vicky. I'll just share my slides. Can you see them in presentation mode?
Yes.
Thank you. So I'm really happy to have this short time to introduce you to our platform, the Embassy of Good Science. For those of you who don't know us, we are a wiki platform that brings together information about research ethics and research integrity and tries to make this information accessible, understandable, and maybe even enjoyable for the everyday researcher and also educators of researchers. So why do we do this? We're doing this because the policy environment, the guidelines, and the regulations surrounding research are constantly changing. New technologies and methodologies make it difficult for researchers to know what's the correct guidance at any one time. And also researchers are under pressure to publish and obtain grants. And there's also this increasing drive for participatory and stakeholder-driven research. So in this complex environment, sometimes difficult to find the right information when you need it. And there are a lot of projects and websites out there that have really useful information for researchers, but sometimes it is hidden in PDFs, or it's difficult to find, or if you actually find it, it's impenetrable for the average researcher. So we, as a group of mostly researchers, came together and received funding from the European Commission to create a website that might change this. We were a European project. We are still carry on with the Embassy of Good Science Foundation, but the project has now finished. And we consulted stakeholders from all over Europe. But our website is global because it's accessible for people from all over the world. So what can you find on the Embassy of Good Science? So I thought for your audience, perhaps the most interesting things are just to talk briefly about the resources and the training, because we know that you are developing both guidelines and training materials. In relation to resources, we as a consortium collected all sorts of guidelines on research integrity and research ethics, national guidelines, but also discipline-specific guidelines from learned societies through systematic reviews, cases to use in teaching, freely available educational resources from all over the world, developed our own scenarios, and we have the ability to make interactive materials, and that I'll talk about a little bit more. So these resources, first of all, were developed by the consortium, but they are augmented by our users, the people who come to our platform, because it's a wiki platform. And you'll see that little blue button up at the top that says add. We are probably the most intuitive and easy wiki platform that there is in the world. People just need to log in with their ORCID ID, and then you use our templates to add information. So if you are working on a project and you have any materials you want to share globally, please do come to the embassy. So what was that interactive element I just talked about? So we've recently added the ability to make interactive educational content directly on the embassy. We're using this open source software called H5P. It's been used for many years in the educational world, and we've integrated it so that you can directly make H5P on the embassy, and it is automatically listed amongst our resources. You can make up to 35 different types of interactive activities, and they can be as complicated as an annotated video or as easy as a drag and drop exercise. Why would you want to make those sort of interactive activities on our website? Because then you can make a course. So you can go to the training section, make a module in steps with your interactive exercises, and group your modules into a course. And we have some projects who have already done that. Here's just a little example of some steps with interactive exercises already integrated. So I'm hoping I'm interesting some people and they might want to come to the embassy and also join our community. So on the community page, you can find projects which are related to research ethics and research integrity. And those projects can specify which initiative, any of the resources, any of the content that they've added across the embassy, across the different categories, that it comes from their project. And then they are automatically listed on their project page. So what you actually get is a very, very easy toolbox from your project on the embassy with very little work at all. So I know I'm probably going over my time, so I'll make it quick. Just the take-home message is that you can find guidelines, cases, scenarios, and openly available education materials on the Embassy of Good Science. With the integration of this interactive content, you can create modules and courses, and you can group them, all the modules, into comprehensive courses on the Embassy for free. The H5P content can be embedded in other learning management systems. So you can make something on the embassy and then put it into Canvas or Moodle or whatever your university uses. And with the new initiative sections, projects are incentivized to add relevant output from their projects to the Embassy Wiki and to group this content into a project toolbox. So we're looking forward to seeing that from the Changer project and also any other project of the people who are watching. So thank you, Vicky, for giving us this time.
Thank you very much, Natalie. So we're also looking forward to seeing the outcomes of the Changer project on the Embassy of Good Science. So thank you very much. We do have 5 minutes to answer some of the questions. To Professor Kurz, what is the difference between optogenetics and using light to train brain organoids?
Well, with optogenetics, you can use light to modify gene expression. That can also be done in the brain. To use light to stimulate, let's say, certain areas of the brain or brain organoid, you need a light sensor. And this light sensor can either transmit light to an electrode, and this electrode then transmit a signal to the brain organoid, or what I presented was this kind of optical cups directly connected to the visual cortex in the brain, or kind of visual cortex-like area in the brain organoid, which can react to light. So instead of having in between this light source or the sensor on electrode, you directly transmit the signal to the visual cortex or cortex Anlagen in the brain organoid. So the difference is essentially one is let's say signaling of neural response into the brain, and the other one is a modification of gene expression, which then can also, of course, for example, simulate a light or a light sensory signal, but it's a different mechanism.
Mm-hmm. Thank you. I hope that this answers your question, Konstantin. One question from Professor Kurz to Professor Lavazza is whether the potency to attain sentience or consciousness is already a reason for specific moral status for the brain organoids. And this is similar to human embryos. So, Professor Lavaggia, if you would like to answer this.
Yeah, thank you very much for the question. It's a very relevant question and not an easy one to be answered. So obviously it's quite obvious to consider this analogy with embryos, but I think we need to be cautious in doing a comparison, because we know that embryos have the potentiality to develop in a human being an autonomous and conscious and rational human being. And obviously, we do not know if organoids can even grow or bigger at the stage. And obviously, they cannot get the stage of autonomous entities. They cannot have any glimpse of rationality or consciousness so far. So there is a huge difference in this sense, and I think we do not need to grant more status to organoids so far. But obviously, it depends on the potential development of future organoids and their capacity to be integrated in a system uh, which can be the way in which organoids can communicate, for example, with the external environment and so on. So for now, I think this analogy does not hold.
Thank you. And another question to you is the animals analogy. So what is the difference between a brain organoid capable to develop sentience and a vertebrate animal in terms of their moral status?
Yes, this is another very important question. Very briefly, animals as moral status, obviously, most of them are sentient for sure. Obviously, we do not have the certainty that animals are sentient as we think we are, but we have good evidence for that. And so we still use animals for experiments because we think that their moral status is lower than moral status of humans. And in general, many scholars think that even conscious brain organoids would have a lower moral status than mammals, for example. So they think that we are allowed to use even conscious organoids for experiments. I'm not sure that this is something of general consensus, but this is the attitude of most philosophers and scientists today.
Thank you. And one last question to you as well. Is it possible to grant moral status to brain organoids based on 4E cognition framework? So this is the embodied, embedded, extended, and enactive frameworks without relying on consciousness as the defining criterion? It's a quite specific question about the 4E framework. So feel free to not to answer this.
Just a word on that. If you consider this approach, obviously you are talking about cognition and not consciousness viewed as the capacity of feeling something. And in the criteria are very, very different. And obviously, you can think that rationality or the capacity of having goals is the most important thing to be considered for moral status. And some scholars share this position. I do not subscribe to this idea, but obviously there is a open debate on that. So this is a quite interesting approach to be pursued.
Thank you very much for this. I would really like to thank you all, both our speakers and the participants, for staying late today. At least I got the feeling that it was a very interesting webinar showing how scientific advancements also have an impact on the ethical issues which are raised along with this organoid technology, with this advancement in organoid technology. So thank you very much to our speakers, and we are very much looking to any future collaborations. We will probably get back to you as part of our research activities within Changer. It was an honor to have you here, and thank you also to all the participants again. So thank you very much all.
Thank you.
Thank you.
So Faye, there is a small question regarding the presentations. The webinar has been recorded, so you will be able to find the recording available. And before I forget, I would also like to thank the communications team from the Changer project. This is Faye Pichilidou and Elena Galifianaki for their support for the organizational part of this webinar. So thank you very much, and have a good evening, everybody.